Pathway analysis. Pathway analysis is the summarization of the measurements of related analytes into one integrated metric of pathway activity. Frequently, cellular activity is conducted by not just individual molecules in the cell, but multiple different molecules that are involved in the similar task. And this create situations where we want to measure not just the individual analytes, but rather the activity of them all. For instance, here in the example below, we have ubiquitin-dependent degradation of cyclin D. This is a pathway described in the literature. It sort of involves multiple different both proteins and metabolites, and actually even protein complexes like the 26X proteasome. Rather than measuring the individual proteins and metabolites of activity, it makes sense to measure these in concert. A potential goal of an analysis of this pathway could be to try to find if there's a difference in activity of the pathway between two different experimental conditions. So what's a pathway? The name seems to suggest that this is a term exclusive for metabolic pathways. However, this is not the way that I will use it, and it's probably not as well what you see in the literature. Actually, we often mean it's just that they are analyzed that in some way are co-regulated. This could involve other situations than just metabolic networks. It could involve proteins that are sitting in the same protein complex. These are that could constitute a pathway. It could be transcripts that stem from genes that are regulated by the same transcription factors. It could also be proteins that sit in the same cellular compartment and not necessarily even interacting with each other. There are multiple reasons why we would like to do pathway analysis. In general, most genes and proteins are acting in concert, and hence it makes much more sense to measure the activity on group level instead of the activity level of individual proteins or genes or transcripts. However, another more subtle one is that in general, and this is true not just for biology, if you find covariation between multiple different signals, is often a much better signal than the individual signal by itself. The fact that you've seen 3 different incidents pointing in the same direction means is a much stronger evidence for something than that you just see one single indication of something. And a third more subtle statistical property of pathway analysis is that when you increase the number of hypotheses of an experiment, you as well induce an uncertainty in your experiment. The more things you test, the more things you have to compensate for as well. So by making fewer hypotheses, like measuring the 1,000 different pathways in reactome, rather than measuring the activity of 20,000 different genes, that leads you to a better signal-to-noise ratio, at least in theory. There are multiple different pathway databases with their different definitions of pathways. We have metabolic pathways definitions like KEGG, reactome. We got the pathway commons. All these 3 are actually instances that contain information about proteins as well as metabolites. So these can be great sources of information if you want to integrate data from, say, proteomics or transcriptomics experiments with the metabolomics experiments. We as well have different interaction databases. We got Quorum, which is a database of known protein complex, often observed in 3D structures. We as well have the interact database and the string database. The string database as well is to a large degree built up with bioinformatical predictions of interactions. And then we got the special case as well of the gene ontology database, and they operate on gene product level, where different annotators have annotated different gene products into their molecular function, as well as their cellular component, as well as their biological process. So it actually contains 3 different parts there. So if you want to know which proteins that are in the mitochondrion, for instance, that's the cellular component, and then you look on the cellular component part of this database. There are multiple methods to summarize activity of analytes into pathway activity. The probably most common one is so-called overrepresentation analysis, or ORIA. This involves conducting a traditional gene or differential expression analysis. So we first look on the list of genes that are differentially expressed. This involves setting a threshold for the differential expression, like a false discovery rate of say 1% or 10 to the minus 10 or something else. Then we take this list of genes and compare it to the list of proteins in the pathway. And we investigate if the overlap between those 2 different lists are larger than expected by chance. So is the ratio between the proteins in the pathway to all measured proteins, is that approximately the same as the overlap between the 2 lists divided by the number of differentially expressed proteins? This could be tested by a hypergeometric test or a Fisher's exact test. There are several tools that can do such tests for you out on the net. Probably most well known is the David tool. In the exercise later on we will use Enrich, another such tool. Another popular tool to do pathway activity measurements are so-called gene set enrichment analysis. Here there's no need of initial threshold for determining which genes that are differentially expressed and which are not. Instead, we look on the full distribution of all the gene products and put in one list. And then we compare that list to the distribution of the samples that are sitting in the pathway itself. So we can now compare and see if these 2 distributions look different. There are multiple different tests to see if 2 different samples stem from the same population or not. Probably most well known is the Kolmogorov-Smirnov test. However, this is seldom used here. Instead, the authors of GISEA, the most popular gene set enrichment analysis tool, they rely on different simulations, so they do different permutation tests instead. We'll use an example of a GISEA implementation as well in our notebook later on.